Somewhere off the coast, a research vessel drifts on calm water, engines silent, and a scientist leans over the railing holding nothing more than an empty plastic bottle. She dips it beneath the surface, fills it with seawater, and seals the cap. To anyone watching, it looks like nothing, just water. But inside that bottle is something extraordinary. An invisible record of every living thing that has passed through this patch of ocean in the last few hours. Fish, plankton, seabirds that dipped their feet in for a moment. And somewhere in that swirl of salt and silence, fragments of a creature that never even touched the boat, never came within sight of a human eye. A dolphin. Its genetic signature, floating free in the water, waiting to be read like a message in a bottle written not in ink, but in the oldest language on Earth. This is not science fiction. This is a real and rapidly growing field of genetics, and it is quietly rewriting what we thought was possible when it comes to finding life without ever seeing it. To understand how something as fleeting as ocean water can hold the fingerprint of a dolphin, we have to go back to the very thing that makes every living creature who it is, a molecule so small that a single teaspoon of seawater can contain billions of its fragments, invisible without extraordinary technology, yet powerful enough to identify a species with almost perfect certainty. DNA, deoxyribonucleic acid, two spiraling strands twisted together like a ladder, holding a code written in just 4 letters, A, T, C, and G, that has been copying itself, mutating, adapting, and surviving for roughly 3.5 billion years. Every organism that has ever lived on this planet, from the first single-celled microbes in ancient oceans to the blue whale gliding through today's seas, carries the same basic code. And every single day, all of them are shedding pieces of it into the world around them without even realizing it. Skin cells, mucus, waste, scales, a stray hair, a flake of tissue too small to see. This constant, invisible shedding happens to every living thing all the time, everywhere. Scientists have a name for the genetic material an organism leaves behind in its environment, environmental DNA, or eDNA for short. And what researchers have discovered about it in the last 2 decades has completely upended the traditional rules of wildlife science. For most of the history of biology, if you wanted to know whether an animal lived somewhere, you had to see it, track it, trap it, tag it. You needed eyes on the creature, or at the very least, evidence of its passage, a footprint, a nest, a carcass. But eDNA changes the entire premise. You no longer need to see the animal at all, you just need to test the water it swam through. Here is where the story gets almost hard to believe. When a dolphin swims through the ocean, it is constantly losing tiny amounts of genetic material. Sloughed skin cells, mucus from its blowhole, even genetic traces in its exhaled breath as it surfaces to breathe. These fragments don't just vanish, they disperse into the surrounding water, drifting on currents, diluting over time, but remaining chemically intact long enough to be captured, filtered, and decoded. Researchers can take a sample of seawater from a location where no dolphin was ever spotted, run it through a specialized filtration and amplification process, and detect, with startling accuracy, that a dolphin had been in that exact area, sometimes hours before the sample was ever collected. It is, in a very real sense, reading the ghost of an animal that has already swum away. The technique itself sounds almost deceptively simple, but the science behind it is a masterpiece of modern molecular biology. First, researchers collect water samples, often just a few hundred milliliters, and pass them through an extremely fine filter designed to trap microscopic particles, including any loose cellular material floating in the water. Then comes the truly remarkable part. Using a process called polymerase chain reaction, or PCR, scientists take the tiny, degraded fragments of DNA caught in that filter and dramatically amplify them, copying specific sequences over and over, sometimes billions of times, until there's enough genetic material to analyze. PCR itself is one of the most transformative inventions in the history of biology, developed in the year 1980s by a scientist named Kary Mullis, who reportedly conceived the idea during a late-night drive along a California highway. At the time, most of his colleagues doubted it could work at all. The notion, like alchemy. Mullis would go on to win the Nobel Prize for a discovery that many scientists now consider as important to modern biology as the telescope was to astronomy. Without PCR, there would be no eDNA science. There would be no criminal forensics as we know it. There would be no rapid genetic testing, no ancient DNA analysis pulled from fossils tens of thousands of years old. It is the invisible engine driving nearly every genetic breakthrough of the last 4 decades, and it is the same engine allowing scientists today to detect a dolphin's presence from nothing but a splash of ocean water. But amplifying the DNA is only half the story. Once scientists have enough genetic material to work with, they need to determine exactly what species it came from. This is where DNA's sequencing enters the picture, Another towering achievement of modern science, one that traces its roots back to a British biochemist named Frederick Sanger, who, in the 1970s, developed a method for reading the precise order of genetic letters in a strand of DNA. Sanger's technique was so revolutionary that it earned him a second Nobel Prize, a feat almost unheard of in science. For decades, his method remained the gold standard for sequencing, but it was painstakingly slow. Reading a single gene could take days. Reading an entire genome could take years. And yet, it was this very technology that laid the foundation for one of the most ambitious scientific undertakings in human history, the Human Genome Project, a 13-year international effort that finally decoded, letter by letter, the entire genetic blueprint of a human being. When it was completed in 2003, scientists had mapped more than 3 billion base pairs, revealing that human beings carry somewhere around 20,000 genes, far fewer than many. Researchers had originally predicted it was a discovery that reshaped how we understand what makes us human, and it proved that a project once dismissed as impossibly large in scope could actually be completed, a lesson that would embolden scientists to attempt even more audacious genetic undertakings in the years that followed. Sequencing technology has since evolved at a staggering pace. What once took over a decade and billions of dollars can now, for certain applications, be done in hours for a fraction of the cost. Modern high-throughput sequencing machines can read millions of DNA fragments simultaneously, comparing each one against enormous genetic reference libraries that catalog the known DNA signatures of thousands of species. This is precisely the technology researchers rely on when they test ocean water for traces of dolphin DNA. The fragments captured in a seawater sample are sequenced and then computationally matched against reference databases containing genetic markers unique to different dolphin species, a short stretch of DNA, sometimes just a few hundred base pairs long, that acts almost like a genetic barcode, distinct enough to separate a bottlenose dolphin from a spinner dolphin, or a common dolphin from a much rarer, more elusive species that scientists might otherwise never have confirmed was present in that stretch of ocean at all. And this is where the story moves from remarkable to genuinely urgent. Because for decades, marine biologists trying to study dolphin populations faced an enormous, often heartbreaking challenge. Dolphins are fast, intelligent, wide-ranging animals that spend the vast majority of their lives beneath the surface, far from human observation. Traditional survey methods required boats, trained observers, hydrophones to listen for echolocation clicks, and enormous amounts of luck. Entire populations of rare or endangered dolphin species could exist in a region for years without a single confirmed sighting, simply because no research vessel happened to be in the right place at the right moment. Some species have become so rare, change that reality in a way that feels almost miraculous. Instead of needing to physically encounter an animal, researchers can now sail through a region, collect water samples at regular intervals, and later determine, sometimes months afterward, once the samples are processed in a lab, whether a target species was present in that water at all. In several documented cases, scientists using eDNA techniques have detected the presence of rare or cryptic dolphin and porpoise species in areas where visual surveys had failed for years to confirm their existence, essentially proving a population was alive and using a habitat without a single animal ever being seen. Perhaps even more astonishing is what this technology reveals about the resilience and traceability of DNA itself. Consider what genetic material actually has to survive to be detectable in this way. Seawater is not exactly a gentle environment for a fragile molecule. It is filled with salt, ultraviolet radiation from the sun, bacteria eager to break down any organic material they encounter, in constant mixing from waves and currents that dilute concentrations further with every passing hour. And yet, DNA, this delicate double helix, persists long enough in that chaotic environment to be captured, amplified, and read with precision. Studies have shown that under certain conditions, environmental DNA in marine environments can remain detectable for anywhere from several hours to a few days, depending on temperature, sunlight exposure, and water movement, giving researchers a genuine functioning window into recent animal activity in a given location. It is a sobering reminder of just how durable the code of life actually is, built to survive far beyond the death or departure of the organism that carries it. This same durability is what allows scientists studying ancient DNA to extract genetic material from fossils tens of thousands of years old, from the frozen remains of woolly mammoths pulled out of Siberian permafrost, from the bones of Neanderthals who vanished from the Earth roughly 40,000 years ago. The molecule that lets us detect a living dolphin from a splash of seawater is the very same molecule that has allowed scientists to reconstruct the genome of an extinct human relative, revealing that most people of non-African descent today carry a small percentage of Neanderthal DNA still coursing through their bodies, a genetic echo of interbreeding that occurred tens of thousands of years ago, discovered not through fossils. alone, but through the meticulous reading of degraded ancient genetic fragments, not unlike the fragile traces drifting through ocean water today. It raises an almost unsettling question. If DNA can persist and be read from water, from soil, from ancient bone, from the air itself in some recent experimental studies, then in a very real sense, every environment on Earth is a kind of archive, quietly recording the passage of every living thing that moves through it. Forests hold the genetic echoes of animals that passed through days earlier. Riverbeds carry the DNA of fish that spawned upstream. Even a single scoop of soil can reveal an entire hidden ecosystem of organisms too, small or too rare to ever be seen directly. Researchers have begun using eDNA to monitor endangered species around the world, from elusive big cats prowling through dense jungle to sharks patrolling coral reefs. And in the case of dolphins, the technique has become an invaluable non-invasive tool for a species that is notoriously difficult to study, using conventional methods. One that doesn't require chasing an animal, capturing it, or disturbing its behavior in any way. The dolphin never even knows it has been found. This kind of quiet genetic surveillance carries enormous conservation implications, especially for the more vulnerable dolphin species scattered across the world's oceans and river systems. Consider the case of river dolphins, some of the rarest and most endangered mammals on the planet, found in murky, sediment-choked waters where visual identification is almost impossible. In these environments, eDNA sampling has become a genuine lifeline for conservation biologists, allowing them to monitor population presence and movement patterns without needing clear water or direct sightings, both of which are often simply unavailable in these habitats. Every water sample becomes a data point. Every data point becomes part of a larger map, slowly revealing where these animals still exist, where they have vanished from historic ranges, and where conservation efforts might make the difference between survival and extinction. For a species that cannot be counted through satellite imagery like elephants on open plains, or tracked with the same ease as land mammals, genetic detection in water may be the single most important tool scientists have for understanding whether these populations are stable, declining, or already gone. But the implications of this technology reach even further than conservation, stretching into territory that touches on some of the most profound and controversial questions in modern genetics. Because if scientists can identify a species from fragmentary DNA recovered from an environment, the same underlying principles apply to identifying individuals. And this is where environmental genetics begins to intersect with an entirely different field that has captivated public imagination for decades: forensic science. The same PCR amplification and sequencing techniques used to detect a dolphin from seawater are conceptually related to the methods forensic scientists use to identify human suspects from a single strand of hair or a trace of saliva left at a crime scene, or the methods used in DNA paternity testing, ancestry analysis, and increasingly in solving decades-old cold cases through genetic genealogy databases. The astonishing capability to extract meaningful, identifiable information from vanishingly small amounts of genetic material has quietly transformed nearly every field it has touched, from wildlife biology to criminal justice to medicine, all built on the same foundational understanding of how to detect, amplify, and interpret the code hidden in every living cell. And this leads to an even deeper question, one that sits at the heart of modern genetics, and grows more urgent with every passing year. If we can now read an animal's presence from a few molecules drifting in seawater, what does that tell us about the sheer density of genetic information saturating the world around us at every moment? Every breath you take releases trace amounts of your own DNA into the air. Every surface you touch, every chair you sit in. Scientists have begun exploring whether airborne environmental DNA could eventually be used to monitor entire ecosystems from a single sample of air, detecting the presence of birds, insects, and mammals moving through a landscape without a single visual observation. It is early-stage research filled with technical challenges, but it points toward a future where entire biodiversity surveys might be conducted almost passively, using sensors that simply breathe in the surrounding environment and analyze what drifts past. For an ocean planet like ours, where roughly 70% of the Earth's surface is covered in water, largely inaccessible to direct human observation, this could represent one of the most significant advances in marine science in generations. A way to finally understand which species live where in real time without disturbing a single living creature. Meanwhile, the field of personalized medicine has been quietly transforming healthcare in ways most people encounter without even realizing the genetic science working behind the scenes. Genetic testing can now reveal whether a patient carries mutations that dramatically increase their risk for certain cancers, allowing for earlier screening and, in some cases, preventive interventions that save lives long before disease ever develops. Pharmacogenomics, the study of how an individual's genetic makeup affects their response to medication, is helping doctors move away from a one-size-fits-all approach to prescriptions, tailoring treatments based on how a patient's unique genetic code processes specific drugs, reducing dangerous side effects and improving outcomes. And running parallel to all of this is the rapidly expanding field of epigenetics, a branch of genetics that has revealed something almost startling, that your DNA sequence alone does not fully determine your fate. Chemical modifications that attach to DNA, influenced by diet, stress, environment, and even the experiences of previous generations, can turn genes on or off without altering the underlying genetic code itself. And in some documented cases, these modifications can be passed down to offspring, suggesting that the experiences of a grandparent might leave a measurable molecular trace on the biology of grandchildren they will never meet. It is a discovery that has forced scientists to reconsider the old, simplistic notion that genes are destiny, revealing instead a far more intricate dance between the code we inherit and the lives we actually live. Stepping back from the laboratory and returning to the open ocean, all of these threads begin to weave together into something remarkable. The same molecular machinery that allows scientists to identify a dolphin from a bottle of seawater is part of an unbroken chain of discovery stretching back to Sanger, reading the first genetic sequences by hand, to Mullis dreaming up PCR on a midnight drive, to the thousands of researchers who spent 13 years mapping the entire human genome letter by letter, to Doudna and Charpentier unlocking a bacterial defense system and turning it into a tool capable of rewriting the code of life itself. Each discovery built upon the last, each one seemingly impossible until someone proved it wasn't, and each one converging, improbably, on a research vessel drifting quietly over open water, where a scientist dips an empty bottle beneath the surface and captures, without ever laying eyes on the animal itself, undeniable proof that a dolphin was there. There is something profoundly moving about that image once you understand what it truly represents. For all of human history, our knowledge of the natural world has been built on what we could see, touch, and directly observe. Now, for the first time, we are learning to read the world through what remains invisible. The molecular residue of life itself, drifting through water, soil, and air, waiting patiently to be decoded. Every dolphin swimming through the deep blue distance of the open ocean is, without knowing it, writing its own presence into the world around it, leaving behind an unbroken trail of genetic evidence that scientists are only now learning to fully read. It is a quiet, humbling reminder that life leaves traces far beyond what the eye can perceive, that the boundary between an organism and its environment is far less solid than we once believed, and that the ocean itself, vast and largely unseen, is not empty or silent at all. It is full of stories, written not in language, but in the ancient four-letter code that has connected every living thing on this planet since life first began. And as this technology continues to advance, growing more sensitive, more precise, and more widely accessible with every passing year, we are moving toward a future where the ocean's secrets will no longer stay hidden beneath the waves. They will simply be waiting, quietly, in the next bottle of seawater someone thinks to collect.